So good morning everyone. So sa continuation sa akong report are the advantages as well as the disadvantages. So yung main advantage is that sa kato po ako na report last time is that compared to the traditional methods of gene sequencing or genome sequencing, um, this next generation sequencing is offers a lot more time efficient and kuan ka ng mas cost friendly po siya kay um, 'di ba atong katong mga traditional na methods kay it will take for about several years, umabot og 10 years to sequence a whole human genome, whereas sa next gen sequencing within a day maka-sequence na siya og one whole human genome, and muto mas kuan po siya mas lesser ihang cost. And wait, kani nga advantage nga fast siya and kuan ni ni naka-gain siya og murag advantage sa kaning a study from katong sa usa ra Japan sa one of the katong company na ni nga tri nga kaning gigamit nila nga sequencing is ang NGS katong Illumina, which ni ni kuan siya sa study atong sa UK wide genome project, which kuan katong ni sequence sila og almost 100,000 and 100,000 genome project, and ilan ni siyang gi-relate atong sa COVID-19, which data from ilang gi-sequence ang data for sa UK ni siya nga nga UK wide COVID-19 project. So ni sequence sila og ilang compare ang genomes of 20,000 patients across the UK who have been admitted kuan katong who have been critically ill with COVID-19, and ni kuha po sila og mild to moderate na effect sa COVID-19, which naabot og 15,000 na genome sequences from patients. So kuan in a short period of time um kato nga project naka sequence sila og 35,000 genomes and with a little cost. So kani nga ang ilang goal ani is to their goal in this um study is to compare those compare ilang gi-compare ang katong severely ill with COVID-19 og katong sa mild to moderate ra nga kuan nakuha sa patients, and ilang gi-compare ang kanang nga genome to have an uncovering genetic factors in susceptibility to COVID-19. So if ilang gikuan if which genome is are more susceptible to COVID-19, and so buto siya ang usa sa mga advantages as well as the related study. And ang iya hapon nga so dili raman ang katong Illumina moy ang company na gamit og source sa sequencing through next gen sequencing. There are a lot more, but Illumina muto siya ang gi-focus kay sa tanan nga na sequence 90% are coming from them. So example sa lain nga sequencing company is Pacific Biosciences, which kani nga different companies offering though next gen sequencing has its own advantages as well as disadvantages depending on their kay dili man sila different companies uses different methods para atong sa ang gi-focus atong unang method which is from Illumina. Yang advantage ato is simple, scalable and high yield. So fast iyahang pag sequence og nakakuha po siya og kuan high yield pud iyahang nakuha. And pero iyang disadvantage ato is it has an expensive equipment. Dili po siya expensive kay kuan ang next gen sequencing itself is kuan a low cost man siya nga kuan pero compared siya sa kuan pero naay sa uban na companies like sa Ion Torrent na company which also uses next gen sequencing. Iyang advantage ato is low cost and it is mas faster pa siya compared sa Illumina na pag-sequence. Pero iyang disadvantage is that it has high rate of sequencing error. So dili pud siya depending on what company dili pud siya overall sa next gen sequencing nga makaproduce og high yield and high quality sequences kay na may uban nga company nga low cost tapos mas fast pero their rate of sequencing can 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 be in high sequencing error and so mura to siya.
Okay, so those are some of sorry, you have mentioned different studies related to the technique that was assigned to you. Actually, this method is considered to be uh, an old method already since there are also some modern techniques that are widely used right now. So I have read some of your article critique, and I'm happy that some have indicated modern techniques since the article is published or was published last 2015. So it's quite old already. So 6 years have passed, and there were so many modern techniques that were already established. Okay, although some of these techniques also have their own respective drawbacks. Now, thank you for that, Mr. Gador. Let's continue with our uh, with the presentation, article presentation of Miss Tubil. You may start, Miss Tubil. By the way, maklaro ra kung tingo guys before we continue.
Yes, ma'am.
Okay, good.
Hello, makita na guys?
Yes, Miss Tubil, you may continue.
Good morning, ma'am. Good morning, everyone. I'm here to discuss about the article about discovery of microbial natural products by activation of silent biosynthetic gene cluster or BGC by Rutledge et al. So the flow of the report is it.
Who wrote, excuse me, Miss Tubil,
Ah,
there are
yes,
only
ma'am.
two authors, so you
Ah,
don't need to use
i-
it out. Maybe you
okay,
can
ma'am.
use Chalice.
Rutledge and Chalice. So the introduction, the flow of the report is about introduction, the objective, the discovery of natural products, appro- the approaches being used, methods for metabolite identification, summary, and future perspectives. So, introduction. Microbial natural products are widely used in
Excuse
medicine
me,
for
Miss Tubil.
yes, ma'am.
Excuse me, Miss Tubil. Pwedeng imo na siyang i-kuan slideshow.
Yes, ma'am.
It's very small, especially for those who are using their phone screens.
So, let's go back. Introduction. Microbial natural products are wide
Excuse me,
are
Miss Tubil. Is it
yes, ma'am.
my screen or it's also the same with your screen, guys? Wala siya naka-slideshow. Maybe you can share your screen. You share your entire screen instead of, by the way, naka-slideshow sa inyo nga and
Wala, ma'am.
say
Wala. Fat.
like,
Wala, wait lang.
may
Try na
say,
to, Tog. Yes, Fat.
kuan sa kay wala siya naka full screen sa amo nga and
Wala, Japon?
wala, Miss Tubil, maybe you can stop sharing the screen usa and then share your entire screen instead of just a particular, is that window or yes, you share na lang your entire screen. That's what I did last time.
Kana, ma'am?
Wala sa ako ang nga side. Share your entire screen. Murag wala man kay gi-share.
I-present now sa chat.
Okay. Sorry.
Naa na, ma'am?
Sige, sige daw. Wala man dire. Nasa inyo guys.
Ay, di pa siya makita. Wait lang, ma'am. Kana ma, makita na?
Yes. Okay na, Miss Tuvil.
Ah, yes, ma'am. So, introduction. Microbial natural products are widely used in medicine for treatment and are also used in agriculture. The rise of resistance to antibiotics is one of the major threats nowadays. Due to the resistance, there is an urgent need to discover new antibiotics. Given the remarkable success of microbial metabolites as starting points for the development of such products, there has been a resurgence of interest in the mining of microorganisms for brand new leads. So, this is an this figure shows an example of drugs developed from microbial specialized metabolites or synthetic analogs since the turn of the century to treat emerging health threats. So the first figure is uh, daptomycin, which is produced by Streptomyces rhosphoros, was developed to tackle the emerging threat of methicillin resistant Staphylococcus eriosus (MRS or MRSA) and another multi-drug resistant gram-positive pathogenic bacteria. So this is figure A shows daptomycin to treat life-threatening infection caused by MRSA. So the next example for of drugs developed from microbial specialized is fedasomycin, which is used to combat gastrointestinal infection by Clostridium Clostridium difficile. Then the third one is the the third example of drugs developed from microbial specialized metabolites is the carfentanil, which is a cancer chemotherapy for the treatment of relapse or refractory multiple myeloma. So, however, traditional bioactivity-based screening of extract from culture of microorganisms that are recognized as prolific producers of bioactive natural products such as actinobacteria and filamentous fungi has yielded disappointing returns owing to the tendency of these approaches to rediscover known metabolites. So the objective of this is this review summarizes the main concepts underlining the genomic-driven discovery of microbial natural products and discuss the pleiotropic and pathway-specific approach that have been developed to induce the production of metabolites that are not expressed under conventional laboratory culture conditions. The researchers also briefly describe the methods used for the characterization of new lead compounds to include with a consideration of future prospects for the field. So the next is genomic driven discovery of natural products. So total genome sequence of actinobacteria and filamentous fungi started to reveal in the early 2000s that they have been much greater ability to generate structurally complex specialized metabolites than suggested by traditional bioactivity based screening approach. So, for example, is the Streptomyces collicolor A32. These bacteria are important and were sequenced because
Mr.
of the
Bill,
yes, ma'am.
please fix on how you read scientific names. Um, it's not the first time. Okay, the Streptomyces silicolor. Okay,
Silicolor, ma'am.
yes.
Sorry,
I mean,
ma'am.
I think our use as what was that? I
Yes, ma'am.
forgot. Anyway, please fix that, Miss Tubil, ha? That's very important in biology and how you read scientific names. If you cannot read it, then maybe you can search on how to read the scientific names.
Yes, ma'am. Sorry, ma'am. So, uh, for example, is the Streptomyces silicolor A32 or these bacteria are important and were sequenced because of their adaptability to adaptability to environmental stress, and source of bioactive molecules for medicine and industry, and rel- and relation to human pathogens. So, biosynthetic gene clusters or BGC for uh, six group of structure structurally distinct specialized metabolites had been identified using molecular genetics approach. However, analysis of the complete genome sequence i- identified 16 additional gene clusters that encode enzymes typically involved in specialized metabolite biosynthesis. So these are example of
Miss Tubil, I have emphasized this before, and I have to re-emphasize this today or this morning. When you make your PowerPoint slides, very wordy, maybe you can just enumerate these enzymes and give short phrases or sentences, salient points on what or how are these enzymes important or what are their functions. Maybe you can just explain those things that you place there on your slide because we can just read it. Okay. And then if I were the audience, I would not prefer to look at slide yung dagkang kaayong words and I don't know where to start. I cannot get the point. Okay? Kay puros words. Wordy ra kaayo. Before unta mahuman ang semester ha, usba ang style sa imong paggama sa PowerPoint.
yes, ma'am.
Okay. Go on.
So the first enzyme is the non-ribosomal peptide synthesis or NRPS. This is the these NRPS are a family of modular multi-enzyme that catalyzes biosynthesis of many specialized peptide metabolites via a ribosome and mRNA independent mechanism. So the
What
next
does it mean? So what does it mean?
So so non-ribosomal peptide synthesis is an enzyme that catalyzes biosynthesis of many specialized peptides.
And then,
They synthesize important peptide products from they synthesize um, they are
sige, continue, Miss Tubel. Let's see if unsa na imong presentation.
yes, ma'am. So the next is the poly polyketoide synthesis. Multi it is a multi-enzyme complexes that are responsible for biosynthesis of polyketides. Then the third is the terpene synthesis is the enzyme that catalyzes the formation of terpene natural products. Then lastly is the NRPS independent is a family of ATP dependent enzymes that catalyzes biosynthesis of side pores without the involvement of
See, you're not even sure if CEDR4 synthetases are part of the enzyme complex or not. Wala nakabold. Is that part?
ay oh part siya ma'am. Sorry ma'am.
Go on.
So identification of BGs, biosynthetic gene clusters or BGCs. So one of the most widely used computational tool in for identifying BGC is the anti-smash or or the antibiotics and secondary metabolite analysis shell. It is a web-based program that identifies biosynthetic gene clusters in an, auto, auto, annotated DNA sequence assemblies. So this photo is an example of general workflow of anti-smash analysis of bacterial, fungal, and plant genomes. And the summary of the approaches that have been used to activate silent biosynthetic gene cluster. So this is the this figure is
Miss Tubel, you did not include fragment-based design and HTS approach in your presentation.
wala ma'am.
Ano man? May I see your slides, Miss Tubil, before we continue?
Eto, ma'am.
Improve your PowerPoint presentation, Miss Tubil.
Yes, ma'am.
You have to report again next meeting.
Yes, ma'am.
Can you see my presentation, guys?
Yes, ma'am.
Okay, so let's discuss chromosomal basis of inheritance. So this is the second part of our discussion. Okay, this can be found on chapter 15 of Campbell. So although this can also be found on some other genetic books, but I preferred the presentation of Campbell for this chapter. Okay, so according to Gregor Mendel, genes were hereditary factors. So before they discovered genes, Mendel have always mentioned already that there are some hereditary factors that are found in the genetic material that are responsible for um, containing different characters. Okay, so these genes are actually located on the chromosomes. Okay, so I believe you know already what's the difference between gene, chromosome, DNA, genome. So I hope as we go on with our discussion, dili na ma-confuse with those different terms. Okay, so that's why the title is chromosomal basis of inheritance, because it is in the chromosome where the hereditary factors responsible for inheritance are found. Okay, so now I believe you have already mastered mitosis and meiosis, since this is very important, okay, in genetics. So mitosis and meiosis were first described in the late 1800s. And so it states that the chromosome theory of inheritance states that genes are found on a specific position within the chromosome. Okay, they have this specific loci. Now, is this true all the time? Is this true all the time? Who wants to answer my question? Hello? Naay, madungog ko or unsa man ni nagputol-putol ko? Hello? Beh, let's start with you, Mr. Montegrejo.
Yes, ma'am. Um, ang
Do you think
unsa
genes
to question, ma'am?
have
Is it
specific?
true? Ah,
Okay, go on, Mr. Montegrejo.
specific siya na siya specific location sa chromosome, ma'am, mo question?
Yes, is that statement true all the time?
Yes, ma'am.
How about you, Mr. Banagudos? Do you agree with the statement? Hello, Mr. Banagudos? Hmm? Wa ko'y nadunggan. Was that a yes? How about you, Miss Triplet? Okay, yes, ang answer di ay. How about you, Miss Triplet? Mendelian genes have specific, no, let's have genes have specific positions on chromosomes. Not Mendelian genes. It's different, totally different when you say Mendelian genes and genes alone. Okay, let's say genes have specific positions on chromosomes. Is that true all the time, Miss Triplet? Miss Triplet? Are you with us, Miss Triplet? Ano man eh, silent man kaayo.
Yes, ma'am.
Okay. All right. So when you say Mendelian genes, it refers to the genes that were used during the experiment of Gregor Mendel in using pisum sativum. Genes don't have, not all genes have um, permanent, although yeah, they have specific position, but these position are not always permanent. Okay, that's in the case for some genes, including jumping genes. No, excluding jumping genes. Okay, so jumping genes really do not have these permanent positions. They tend to move around the chromosome. That's why they are called as jumping genes. Although these genes will be discussed later on as we go along our lecture. So chromosomes also undergo segregation and independent assortment. These are also known as law of segregation and law of independent assortment according to Mendel. Okay. Then the behavior of chromosomes during meiosis can account for the different laws of Mendel. Okay. For example, this one. So you can see here, P generation. P refers to what? It's parental generation. And then we have here F1 generation or first filial generation and second filial generation. Okay. So in this case, it's using a cell. What's the genome size of this cell? Based on the number of chromosomes. That's 4. Okay? That's 4 chromosomes. 4, is it okay, wala gi-specify if it's male or female. Anyway, so one is male, one is female. So both of these cells contain 4 chromosomes. So here, YR refers to the genes that code for the yellow and round seeds of this particular plant species. As for the other parent, it contains recessive alleles. By the way, what are alleles? What are alleles? Let's go to Miss Elnas.
Each copy of the gene from each parent.
Copy
I
of,
dip.
hmm?
Yeah.
Ano to? Ano to? It
And
is,
variant ay ma'am. Variant form sa
okay.
gene.
Can you give an example, Miss Elnas? Can you give an example? How about you, Mr. Gador? Can you give an example of alleles? So
Anong
in
color
this
sa air ma'am?
color sa
Ay.
or let's say here sa kaning example nako sa PowerPoint presentation.
And wrinkled o ganyan. Wrinkled and round.
All right. Wrinkled and round, and the other one is yellow and green for color. Okay, so those when you say alleles, those are the alternative form of a gene. So for this example, we have alleles yellow and green for the color of the seed, and then round and wrinkled for the texture of the seed, shape of the seed. Okay. So right after meiosis, it produces half of the total or haploid, one set of chromosome. Okay. So instead of producing 4 chromosomes, since nga undergo siya meiosis, it refers both meiosis 1 and 2, okay? So, produce na dahil siya haploid or gametes which contains haploid, one set of the chromosome. And then fertilization happens, and then it produces offsprings. So young offsprings, all offsprings produce yellow and round seeds. Okay? And then, undergo na po siya meiosis to produce gametes. Okay, this is where the laws or Mendel's laws come in. Okay, so we have law of segregation, which states that the two alleles for each gene separate during gamete formation. And then law of independent assortment states that alleles of genes on non-homologous chromosomes assort independently during gamete formation. All right. So for this example, okay. So during metaphase, what are the remarkable events? Can you still remember the remarkable events during metaphase? Okay. The chromosomes align at the metaphase plate. Okay, so sa first law niya, the two alleles for each gene separate during gamete formation, which means each of the alleles will separate. Okay? According to law of independent assortment, there is no pattern. Okay? There is no pattern to the arrangement of these alleles, especially on non-homologous chromosomes. Okay? So, naatay arrangement here nga ang tungod sa imuhang homologous chromosome is both dominant. Whereas in this in this cell, ang tungod sa imuhang dominant Y is recessive R. Okay? These are non-homologous chromosomes. But in the case of homologous chromosomes, if this codes for the shape of the seed, then of course, the chromosome, the homologue also of the seed, be it dominant or recessive. But for non-homologous chromosomes, it doesn't follow any pattern. Okay? Wala siya pattern. Pwede ni siya magsunod puros dominant. Pwede po siya dominant, recessive, or dominant, recessive, recessive. So there is no pattern. Okay? Assortment of these non-homologous chromosomes is random. Okay? So right after anaphase, there will be separation of anaphase 1, separation of homologs. So I hope you can still remember the different remarkable events during meiosis and mitosis. It's very important, especially on meiosis. Okay, so during separation of homologs, okay, it become already haploid. Okay, each of the daughter cells will contain one set of chromosome from the parent cell or relative to the parent cell, but sister chromatids are still intact. Okay, and then meiosis 2 will take place. There will be separation of the sister chromatids. See, the gametes carry different alleles. Okay, so naay gamit nga puros dominant, naay gamit pod nga puros recessive. And there are also gametes that contain both recessive and dominant. Okay. So if we are to cross this different offsprings, it will yield a different ratio, in which case there will be combination sa imuha nga parental characteristics or parental characters. So here, 9, that's 6, 7, Okay, 9/16 or 9 out of 16 offspring will give rise to seeds that are yellow and round. Okay, 3 will give rise to 3 out of 16 offsprings will give rise to seeds that is green in color but round in shape, and so on and so forth. So when you say these characters, these are actually phenotypic characteristics. Okay, when you say genotypic characteristics, you will say or you will write these alleles, dominant and then the recessive one. So this one is actually an example of complete dominance. Good thing during the experiment of Mendel, all of the characters he has used were completely dominant over the other. Okay, so there was no problem during tracing of the different characteristics. Now, I hope you're familiar also with this person or scientist, Thomas Hunt Morgan. He was an embryologist, and then his experiments were more focused on Drosophila melanogaster or fruit flies in order to provide evidence that chromosomes are really the location of the genes or the heritable factors according to Mendel. Okay, so Mendel used plant, Thomas Hunt Morgan used animal. Okay, so he used Drosophila melanogaster because of these reasons. Okay, first they produce many offspring. Okay, and then aside from that, a generation can also be bred every 2 weeks. Ilang life cycle is quite fast. Ilang lifespan is very short. So you can produce generation every 2 weeks. Okay. And then they also have few pairs of chromosomes, are only 4 pairs. So 2n is equal to 8. So it's easier to track. Okay. And mas dali gamiton ang Drosophila melanogaster compared with um, other organisms. Okay, so when you say wild type, these are phenotypes that is the most common in fly populations. So usually they are denoted with superscript plus if it is wild type. Okay, alternative to the wild type are called mutant phenotypes. Okay? So kung unsa gani phenotype nga pinaka-dominant sa population, then that means it's the wild type. Okay? Other than that, it's mutant. For example, this one. Which do you think is the mutant and which do you think is the wild type? Color of the eyes, na red. This is actually brick red, and then there is also white. Okay, so here, brick red color of the eyes of D. melanogaster is the wild type, and then white eyes called mutant. Ay, nasa naman to? So in one experiment, Morgan mated male flies with white eyes, so mutant, and female flies with red eyes. So the first filial generation generated offsprings which have red eyes. However, the second generation showed a 3:1 ratio in which 3 out of 4 offsprings, that's 75% out of 100%, all generated offsprings with red eyes. And then white eye ratio, no, 25% have white eye. Okay. So Morgan determined that the white-eyed mutant allele must be located on the X chromosome. Why? Why? Ano mang nakaingon
Because
siya nga naa sa X chromosome. Yes, Soliera?
white eye ratio kay white eye gi-mention diri sa taas ma'am nga only males had white eyes.
And
So
then,
and kana meaning that ang kuan niya ang mutant allele is located on the X chromosome.
anong dili man Y chromosome? Males also contain Y chromosomes and not females.
Kasi ba pag kuan ma'am, pag female ma'am kay XY.
Hmm?
So since both
Females XY, are you sure?
ay dili ma'am ang kuan di ay if kung ang mutant allele niya is nasa X chromosome, meaning ang if adto sa female is dili siya mahimong white eyes because of katong naa siya katong mas mo dominate man tong sa red
Are you
na
sure?
characteristic niya. Maybe.
Are you
Sa
sure?
father man ang X chromosome, ma'am.
So,
Sa male,
Sa Abril?
sa male fruit flies ang dominant kanang X chromosome sa iyahan ng kuan sa male Drosophila.
Wako kasabot Abril. Sa imong tubag.
Kuan siya ma'am sa
Dominant.
it's located in the X chromosome ma'am because males only have one copy of the X chromosome. So wala sila laing ma-express if
Nya nganong dili man sa Y? Nga naman pud sila
to.
Y.
Does that make sense?
Ngano man wala sa Y?
Dili siya sa Y.
Okay?
Mas dominant
Gamay
man
man
sa
ang Y mo.
kuan.
So if if gamay day ang Y, it's not possible that the gene that codes for the color of the eyes is found on the Y chromosome.
Mas dominant sa
Not
kuan
necessarily.
ma'am ay sa male ang kuan X. Ang
Mas
kana
dominant ang sa male nga X chromosome over the Y chromosome.
ay dili ma'am ang
Why-
gene nga
why-
ma-express para sa white eyes ma'am ba sa male.
Unsa sa to Soliera?
Mas dominant ang gene nga ma-express og white eyes sa male compared sa female.
Really? Any other answer? Why did Morgan concluded that the one that is responsible for generating offsprings with white eyes is found on the X chromosome and not on the Y chromosome, when in fact it's all males that contain white eyes? Why not on the Y chromosome? Yeah, Mr. Montegrejo, you mentioned. Okay, go on, Mr. Banagodos. Huh? Okay, so you think mm, mm. Note that the parents di melanogaster, ang male ang naay white eyes. So, which means that's either of the X or Y chromosome which contains the gene responsible for coding white eyes or the mutant nga allele. Okay, but my question is, why is it that he concluded that it must have been located on the X chromosome and not on the Y chromosome?
Um,
Yes, Mr.
ma'am,
Gadot.
if kung naa siya sa Y, dili siya maka-produce og um, female nga white
Okay.
with white eye.
What's wrong with Y chromosome? Or what's with Y chromosome? How is X chromosome different from Y chromosome? Yeah, Mr. Montegrejo, you've mentioned that Y chromosomes are quite smaller in size than X chromosome. Can you elaborate on that?
Yes, ma'am. As far as I know, Y chromosomes are smaller. Then, ang iyang role is only for the regulation of certain characteristics, particularly sa sex characteristics. So, dili kay siya mo code og other traits. Dayon, yeah. Mura siya og
Okay.
X chromosome siya, ba nga murag mas gamay ni degenerate nga X chromosome. Mura akong nabasahan.
Okay, that's correct. So Y chromosomes are quite smaller in size compared with X chromosomes, and mostly it codes for sex characteristics or male characteristics. Okay, so basically, most of the genes that codes for the different parts of the body are found either in the autosomes or X chromosomes. Although there are also some parts of the Y chromosome that is complementary to the X chromosome, which is really important for pairing during meiosis. Okay? So he concluded that it came from the X chromosome since Y chromosome mostly codes for um, uh, sex characteristics. Okay? So based on his results, Morgan's findings supported the chromosome theory of inheritance. Okay? So this is his experiment. So ang male contains red eyes and females contain males contain white eyes and females contain red eyes, and all of the offspring has red eyes. However, when they cross these offsprings, 25% of the total second filial generation generated males with white eyes. Okay, so and no, sorry, not 25%, but 25% for the whole offspring, but 50% on the male population. Okay, for example, na kay 50% sa imuha nga, na kay 100% male population, half of it, that's 50%, have red eyes and the others have white eyes. So in conclusion, look. So there are different ways in writing mutant and wild type alleles. So in the case of Drosophila melanogaster, they usually use superscript plus for wild type, and then for mutant, it is uh, just the letter. Okay, so wala siya superscript. So in some characters, they usually denote these characters either capital letters or small letters. Okay, so it depends on the organism or test organism being used in the experiment. So here we have here parental generation and then F1 generation. So all of the offsprings coming from the mother produced one type of allele, which is wild type, whereas for the sperm, whereas for the male or paternal gametes, half is normal or wild type and then half is mutant. Okay. So according to law of segregation or law of independent assortment, unsa may ma-apply din he? Law of segregation or law of independent assortment? Which do you think? Same apply din during production of the meats. Hello? Which is the most applicable? Law of independent assortment or law of segregation? Miss Tubil. Miss Tubil? Anyone from the group?
Law of segregation ako, ma'am.
Why?
Kay iyahang inheritable trait or gene controlled by a pair of alleles man, ma'am.
And so?
So, kuan ang iyahang kanang traits like for example sa Drosophila melanogaster is kanang kuan determined by the alleles present on both like for example the first generation nga organism sa kuan Drosophila is kuan man.
Okay, yeah, that's correct. The most appropriate law that should be applied here is law of segregation, since the different alleles on the parent cell is segregated during gamete formation. Say for example, here sa sperm, kay duha man ang iyang alleles, one wild type, the other is mutant. Okay, so ma-separate ni siya or ma-segregate ni siya producing an allele or a sperm cell which contains mutant allele, and then the other sperm contains wild type allele. Okay.
Ma'am, pwede mo ask.
Yes, go ahead.
Kanang what about sa katong kuan, ma'am? Organisms nga mo undergo og sex change, applicable Japon ang number of chromosomes?
Okay, note that not all organisms have XY rule in gender or in sex. Sorry, not gender, but sex. I think I have it here. Yes, this one. Natay gitawag og XY system, natay gitawag og XO system, ZW system, haplodiploid system, and there are also organisms that can undergo sex change like parrotfishes.
Yes.
Okay? So, kaning akong gidiscuss is XY system. Okay?
Okay, ma'am.
And there are
Thank you, ma'am.
also organisms that can undergo sex change, and not sex change, but there are organisms wherein their sex are determined by the environmental factors. Example, reptiles. What determines their sex is temperature. Okay, I hope you're aware with that. So this one is only applicable to XY system. Klaro ra?
Yes, ma'am.
Okay. So sex-linked genes exhibit unique patterns of inheritance. Now, in humans and some other animals, there is a chromosomal basis of sex determination. But for some organisms, there is no chromosomal basis of sex determination because some of these animals or the sex of these animals can be identified by a wide variety of factors other than the chromosome. No problem with humans because we're using XY system. That's why we can also pinpoint certain genetic diseases, okay, and their causes. So here, we've mentioned this a while ago, there is a larger X chromosome and a smaller Y chromosome. So only the ends of Y chromosome have corresponding regions, homologous regions with X chromosome. This is very important for or during um, um, synapsis. Okay, so during synapsis, the different um, parts or the ends of the Y chromosome um, binds or synapse with that of the X chromosome. Okay, this is really important in order to prevent aneuploidy. Okay, tendency man god if dili magtapot ang imong homologous chromosomes, these chromosomes will either be um, lost during the process in meiosis or madoble ang imong chromosome number. Okay, so that's a problem later on. So the SRY gene on the Y chromosome codes for a protein that directs the development of male anatomical features or the male uh, sex characteristics. Okay. So here's a figure. So if we are to compare Y and X chromosome, Y chromosome is relatively smaller compared with the X chromosome. So females again are XX and males are XY. So each of the gametes contain only one sex chromosome. For females, all ova contain X chromosome, while in the male, it could either be an X or a Y chromosome. And then, yeah, I've mentioned other animals have different methods of sex termination. So we have XY systems, XO system, ZW system, haplodiploid system, and there is also another method in sex determination. We have environmental factors. So sa XO system, if it contains only one sex chromosome, and that is X, then it is male. If duha ang iyahang X chromosome, it's female. So tendency ang males contain 24 chromosomes. Females contain 24 chromosomes, male contains only 23 chromosome. That is for XO system or X0 system. For ZW system, for some insects, for some insects. For some avians, what they have is ZW system. So if it's ZW 76, okay, 76 ilahang autosomes plus sex chromosome ZW for females and ZZ for males. For some others, for some other organisms or some other insects, they have haplodiploid system. If ma-fertilized, female. If di ma-fertilized, male. Okay. Sa reptiles, temperature ang ma-identify sa sex or ma-determine sa sex. Okay. So a gene that is located on either sex chromosome is called a sex-linked gene. Okay. So if it's found on the X chromosome, it's X-linked. If it's on Y, it's Y-linked. Okay. That's why if a gene or if a disease is caused by gene that is found on the sex chromosome, we call it as sex-linked disease. Okay. Or Y-linked if it's found on Y, and X-linked if it's on the X chromosome. Okay. All right, so this is the explanation why Thomas Hunt Morgan concluded that white eyes is found on X chromosome because it contains genes for many characters that are unrelated to sex, whereas Y chromosome mainly encodes genes related to sex determination. Okay. X-linked genes follow specific patterns of inheritance. For a recessive X-linked trait to be expressed, females should have both copies. It should be homozygote. Note that even if females contain both X chromosomes, only one X chromosome is expressed and the other one is inactivated. But in the case of males, both X and Y chromosome are expressed. Thereby, if there is a disease or a gene that causes a disease in either of the sex chromosome of females or of males, it's 100% expressed. Okay, unlike sa females. Females can only be carriers, but it also depends on the type of dominance. If it's complete dominance, then 50% pwede siyang ma-express, 15% pwede siyang ma-inactivated. But if it's incomplete dominance, okay, or co-dominance, that's another story. X-linked recessive disorders, also, sa female, as a male, they only need one copy of the allele in order to be expressed. Why? Because they contain both X and Y chromosome. So X-linked recessive disorders are much more common in males than in females because X chromosome is expressed in males but not in females, depending on the chromosome that is expressed. Pwede man siyang inactivated, pwede pong i-express. Example, this one. So, naata dire different parents, process of parents that contains varying tawag ani alleles. Okay. So, which do you think causes the disease? Is it the dominant N or the recessive N? Is it X-linked or Y-linked? Mr. Gador.
Ma'am, unsa to ka dyan?
What causes the disease? The dominant N or the recessive N? Second question, is it X-linked or Y-linked? Answer the first question, Mr. Gador. And then Miss Abril will answer this. Unsa to imong answer? Is
Sa
it X-linked or Y-linked?
Y-link.
Ang
Ay, wait, sa X dahil mo.
Y. Support your answer.
Wait sa ma'am ka dyan. The daughters
Sige.
will have the normal phenotype but will carry the mutant. The daughters, when mother is a colorblind father will transmit the mutant allele to all the daughters.
Okay, sige. While reading, Mr. Gador, let's continue with the answer of a first question. What was my first question? Is it the dominant N or the recessive N? Why not answer my question, Mr. Salapa? Mr. Salapa.
Yes, ma'am. As ay mas dominant ang egg,
No.
ang
Which allele is responsible for the disease? Is it dominant N or recessive N?
mm, ang which ang dominant N, ma'am.
How about you, Mr. Banagodos? Banagodos? Oh, Miss Triplet.
Obviously is the recessive N.
Okay.
Um, all dark orange boxes contain recessive N, unlike the white colored boxes which have the dominant N, which has no color blindness, unlike the dark colored orange boxes.
Okay, that's correct. Okay, let's go back to you, Mr. Gador. Is it X-Link, Y-Link? Hello?
X-link, ma'am.
Okay, it's X-Link, of course. Okay, here's the first letter A, cross. So, ang iyahang mama contains both dominant alleles, but the father contains the recessive allele. Okay, so it expresses the disease. Say, for example, the dark orange one refers to the diseased organism. After after gamete formation, okay, so 50% of the sperm contains, or half of the sperm contains the recessive allele. It is found on the X chromosome, 50% Y. If there is cross, if ang iyahang sperm nga makuha during fertilization, okay, it will produce an offspring, a female offspring, which is a carrier of that particular chromosome. Okay, that's why it's lighter in color. I think this one is colored peach, if I'm correct. I'm not really good with colors. Anyway, lighter ang ihang color because it is a carrier. Whereas here, 50% normal. Okay? 50% normal ang iyahang males. Okay? So, if ang imong i-cross is normal female and a deceased male, 50% of the offsprings, male offsprings in particular, are normal, and the other 50%, or all female offsprings, may be carrier of that recessive allele. Okay, so 50% normal, 50% carrier. All females are carriers, all males are normal. On the second cross, if the male contains normal allele, and then female is carrier, if i-mate nimo ang normal male and carrier female, this will be the result. Okay, so ang result niya, 50% of the male offsprings will be affected by the disease, and the other 50% is normal. In the case of female offsprings, 50% will be carriers, 50% normal. Okay. So may tabuan ni, if mo na iyang offspring, 25% carrier, 25% disease, 50% normal. But if the deceased male is mates with male, what will happen is that you will increase the probability of getting the disease in the offspring or the first filial generation. What happens here is that 50% of male and female offsprings would most likely express the disease, and then 25% of female offsprings, or 50% of female offspring will be carriers, and 50% of male offsprings will be normal. Okay, So this is why consanguineous mating is discouraged, because most likely there will be an increase in the probability of expressing a particular disease. Okay. So some disorders that are caused by recessive alleles found in X chromosomes: color blindness, it's X-linked, and then Duchenne muscular dystrophy and hemophilia are also caused by X-linked um, mutations. Okay, so this one I have mentioned this. So even though females contain both X chromosomes or two X chromosomes, only one is expressed and the other one condenses into a bar body. So we call that as X inactivation. Okay. But if a female is heterozygote for a particular gene located on the X chromosome, and then some of these X chromosomes are expressed and the other X chromosome that contains another allele is also expressed, it will create a mosaic for that particular character. For example, this cat. Okay. So the color of the fur is found on the X chromosome. So if she is heterozygote, sa ato pa, it contains different alleles. So one allele one X chromosome contains the gene that codes for an orange fur. On the other hand, this chromosome contains the allele that codes for black fur. Okay, so some of the orange allele or some of the X chromosome that contains allele that codes for orange fur is inactivated and the others ang black. And then, of course, ang imuhang resulting individual is a mosaic, okay, of both characters. So this part ang na-inactivate ani is the black. Okay, the X chromosome that contains the allele that codes for the black fur. Okay, in contrast to this one. All right. Reports for cell division, you will report mitosis, Ms. Abril. Ako ning gipasunod ha. So ang first nga reporter usually si Elnas, followed by Gador, Banagodos, Montegrejo, Triplet, Tubil, Soliera, Abril. So sorry. So ang last is si Tubil. Mr. Salapa, what is your decision so that I can also include you?
Dili na lang kumo dayon og shift, ma'am.
Okay. So
Yes, ma'am.
sige. So instead of Gador, so sa mitosis, ikaw Abril. Dayong sa meiosis, ikaw Mr. Salapa. Okay?
Sa mitosis ko, ma'am. Sa
Meiosis.
meiosis,
Okay,
okay, ma'am.
sunod na ko ni Abril. Usa ta mo balik ni Elnas. So sa life cycle, ikaw sa Elnas.
Okay, ma'am. Yes, ma'am.
So dire, nakatuyok na ba tayo sa O pa ka-report Soliera, no?
Wala pa, ma'am.
So this one, kasalapan siya.
Alphabetical ma'am.
Wala, ako ra ning girandom. But na naman ko ikuan pasunod. So kani siya ka Elnas.
Gosh.
So kani siya care of Soliera ni imo ni Soliera ha.
Yes, ma'am.
Para ma-checkan nako ang naka-report og article. So far ang naka-report og article ikaw pa og si Tubil.
Karyota, ma'am.
Tubil.
Maayo sa essay banhin, ma'am.
Tubil? Hello, Tubil.
Yes, ma'am. Yes, ma'am.
Magpadayon tas imong report this coming Thursday, ha?
Okay, ma'am.
Okay. Before we proceed with the reporters, there is second lecture. Nakuha ra ninyo, guys?
Yes, ma'am.
Yes, ma'am.
Maybe I will post this article sa Google Classroom later.
Kano sa amin
Question
kung mga
before,
report, ma'am?
andam lang. If sayong mahuman si Tubil, then we'll proceed with the next reporter. Okay? It's coming
Dayon
Thursday.
Saturday ang exam Japon, ma'am.
Yes, Saturday atong exam 10.
Unsa na nga chapter,
Mag-oral.
ma'am?
Atong pinakauna.
Sa ka-Hartwell?
Yes. Atong i-discuss last time. Ito siya. Okay?
Ma'am.
Mao ba sakto ba? Ito. Dayon kani siya next week na ni nga exam. Basta mahuman na tanang report including the article. Okay? So, the same gyapon ang deadline for this critic paper is
1 week.
next week Tuesday. Anyway, I'll just post this sa Google Classroom including the deadline.
Pwede
Okay,
ibalik
Carol?
ako report, ma'am.
Ha? Ana. Wara?
Kaya sa ma'am.
Campbell, Japan ni ma'am.
Ha? Salapa?
Kaya.
Salapa.
Yes, ma'am.
Please study both meiosis 1 and 2, ha? And then compare it with mitosis.
Okay, ma'am.
Okay. Question before we end this call?
Campbell, ra ni ma'am? Or
Bisag unsang libro. Isa mo, ganahan. Wala na yung question?
Wala
Wala
na,
na,
ma'am.
ma'am.
Okay.
Wala naman.
Alright, sige, great. Thank you so much for attending our short lecture. Short kay 11:36 pa man. Okay. I'll see you next meeting Thursday. Okay. Bye. You may now leave this call.
Thank you, ma'am.
Bye, ma'am. Thank you.
ma'am.
Bye,
ma'am.
ma'am.
Thank
Goodbye,
you.
ma'am. Thank you, ma'am.
may
Bye.